Hello again, I'm Steve Eath here. Welcome to this second video on the use of the data mining tools that are present on the Some Breast Cancer Cell Line knowledge base. So hopefully you watched the first video where I basically gave you a tour of the website and how it's organized and the data sets that sort of underlie the data mining tools that we've built to help you understand the cell lines you work with better. So today I want to start by taking you through some of these tools to show you how they work and to make people a little bit more comfortable with them, because what I think you'll find is as you work with these data mining tools, you'll learn a tremendous amount about cell lines you work with and even cell lines that you don't. I guarantee it, because I'm still learning about the cell lines I even developed 20 some odd years ago. So I want to do one video for each data mining tool so that we keep these pretty short. And what I want to do today is start with, I think, the most basic one and the one that I use the most, and I expect that you might as well, and that is the gene query tool. I'm going to go ahead and share my screen now, and we will get started. Okay. So the data mining tools for the 50 breast cancer cell lines, as I showed in the last talk, are under this banner heading. And we can go right down from here to the gene query tool. And you can take a moment to just sort of read through this first paragraph, which talks about how this tool is designed to allow users to pick any gene you're interested in and view both the genomic and functional genomic data for 51 breast cancer cell lines. There's CRISPR data, there's RNAi-based screen data in here. All of the genomic data, copy number, gene expression, point mutation, all of these data are leveraged within this tool. And I want you to note here, as I'll show you, that within the tables that come up, each column is clickable so that you can rank order the data any way you like for what you happen to be most interested in. I want to just point out again that, again, we use the screen data a lot to try and functionalize all of these genomic data. And so because in this particular part of the website, we're using data from the Han Lab and DepMap portal, they use an algorithm called series to analyze their genome scale screen data. And what you should understand is that anything you see that has a series score of -0.5 or less are considered to be hits in the screen. So the more negative the number, the stronger the hit in the screen, the more essential that particular gene is in that cell line. We also have copy number data and gene expression data. The gene expression data is given in log2 space, so this LFC column you're going to see here, that stands for log fold change. It's log2 relative to all breast cancer cell lines in the database. So we basically took the mean expression level for all of the genes across the entire database, and the ratio you'll see in log2 space tells you the relative expression of that gene compared to all other breast cancer cell lines, and the same is true for the DNA amplification part. Okay, so I think this is the most useful tool, or the one that might be used most, because most people who work in breast cancer or would come to a site like this have a gene that they're most interested in. So for my career, I've been very interested in ERB2 and EGFR and others. A lot of people in our field are interested in the estrogen receptor. And so, but you can pick any gene you want, but let's just start with a couple of these common ones to see how this tool works. So to basically start the gene query, you click here and then hit your backspace to make that go away, and then just type in your gene, ESR1. That's the gene name for the estrogen receptor. The database will recognize it, and you click on it, and now the software tool will go to work. And you can see we have 6 pages of data. Now, when this comes up, it will come up rank ordered by the so-called Achilles score. So that's that series score I was telling you about. So the idea here is that the cell lines here are rank ordered by most essential for the estrogen receptor to least essential. And so And we used as a starting point the CRISPR data to do that. So what you see right away is the first cell line that comes up is HCC 1428. Now you see here, as you would expect, right, estrogen receptor in primary breast cancer is never amplified. And so the copy number amplifications are never more than 1, sometimes even a little less than 1. But here the log fold change is showing the relative expression at the message level for ESR1 compared to all breast cancer cell lines. And you can see, as you'd expect, these ER positive lines significantly overexpress the estrogen receptor message, the ESR1 gene. So that this is log fold change. So that means the HCC 1428 cells express 2 times 2 to the 5th times more estrogen receptor than the average, or roughly 32 times higher levels of estrogen receptor. Here's everybody's favorite estrogen receptor positive cell line, MCF7, 2 to the 4th relative expression compared to other cells. And so again, so what this does is it rank orders and shows you right away the cell lines in this database that are considered ER positive. And you might be surprised at how many there are out there. In our world of the literature, what we're used to seeing is data for MCF7 and data for T47D cells. And you probably don't recognize a lot of these cell lines, but there are many others that express very high levels of the estrogen receptor. So these are, again, rank ordered now by the CRISPR score, but we can also rank order them by the RNAi score, or the shRNA score here. Now T47D here, it does appear in this list. And that's because for this cell line, we have RNAi data, based screen data. We don't have CRISPR screen data. So it's always important and interesting to sort of flip back and forth because once again, you see that by rank ordering by the RNAi-based screen, we now find HCC 1500, which has enormous levels of expression of ER at the message level, and again, T47D, and a couple of other cell lines even pop up that you see that are ER positive, CR 7530, UACC 812, that you don't see if you only use the Achilles data. And then by contrast, there are cell lines for which we only have CRISPR data and we don't have RNAi data. So again, if you want to see all of the breast cancer cell lines that are ER positive, use both and click on both in order to rank order the cell lines based on the level of essentialness. Again, the more negative the number, the more essential. So let's explore that a little bit further. And what you see, and you won't be surprised to see, that all of these cell lines that overexpress the estrogen receptor, the ER is an essential gene. So as I said, the cut point here is negative 0.5. And so even this cell line with the lowest relative levels of ER, ZR75, and then KPL1, these are considered hits in the screen. And you can see they were hits in the RNAi screen too. In fact, you see that consistently all the way through for this particular gene. There's great correlation here for the estrogen receptor in the data from the CRISPR screen and the RNAi screen. The levels of sensitivity are a little different, so the numbers may not be as strongly negative in this case, but using this cut point of negative 0.5, you can see that there's great concordance here in terms of which cell lines the estrogen receptor is essential in. And the same is true for these additional lines that we can see when we rank order by RNAi, HCC 1500 quite essential, T47D essential, of course MCF7 essential in both, and so forth. And then as you now work your way through the pages, you will also then notice right away as we get into these cell lines that do not express the estrogen receptor, then the essentialness drops right off. And there, in fact, aren't any cell lines that are ER negative that have series scores less than negative 0.5. So this is a great example to start with. It's very clear cut. We have a bunch of breast cancer cell lines that are ER positive. And in every case, the estrogen receptor is an essential gene for these cells, and a whole bunch of cell lines that are ER negative. And for every case, the estrogen receptor is not found to be essential in the screen. So it's a beautiful starting point, a beautiful example of how we can begin to look now at a genomic feature associated with breast cancer, that is, the overexpression of the estrogen receptor, and begin to relate it to the essentiality of that gene in those cells that have that genomic feature. Again, the estrogen receptor, the correlation is really, really good. If it's overexpressed, it's essential, and you can see that very clearly for each of these cell lines, and you can again rank order them based on essentialness. And I think there's reason to believe, and we could talk about this in a separate blog or a separate post, that these differences in relative essentiality are meaningful and are really associated with the level of overexpression. And that's a pretty interesting feature of this. So again, all of these data come up at a mouse click for a whole panel of cell lines, all the cell lines that you're interested in, that you've known and loved over the years are all here, and you can look at, and you can see now there are a number of lines as we get down to page 5 for which we don't have any screen data. They haven't been screened yet in the Han lab, but we still have the genomic data, and so you still can identify cell lines based on estrogen receptor expression levels. Okay, so that's ESR1. That's an easy one. Let's take another example, a gene that I've spent a lot of my career working on, the ERB2 oncogene. And of course, I should mention here that if you type in your favorite gene and it doesn't come up, I suggest the reason is you're not typing in the actual official Hugo gene name. So go look that up and be sure you have the gene name. So in the case we just looked at, if you type in ER, nothing's going to happen because the gene name for the estrogen receptor is ESR1. In this case, if you were to type in HER2, nothing's going to happen because the official name for that gene is ERB2. So be sure you're always working with the official Hugo gene name for the gene you're interested in. Okay, so here's ERB2. Now this is a gene, as we all know, is activated by a process of gene amplification. And so like all of these, every time you went through a gene, the cell lines are going to come up rank ordered by this CRISPR-based Achilles score. And you can see there's some association here with DNA amplification. And it's a little bit curious. So instead of looking at it from this perspective, let's look at it from the DNA amplification perspective. So now we're rank ordering the cell lines based on DNA amplification. And this is what you'd want to do for this gene because, again, we can identify 10 or 12 cell lines, maybe 14 cell lines with ERB2 amplification, and then it drops off. And the thing you will notice again, and you won't be surprised to find that amplification and overexpression go together. All of these cases where there's significant copy number gain, focal amplification, ERB2 is overexpressed. Goes all the way down, and now when we get away from the amplified genes, the expression levels sort of go down as well. So now we have a way to look at ERB2 in a whole panel of cell lines and looking at these lines with respect to the level of gene amplification and gene overexpression. Now, as I said, for the ESR1 gene, you may be surprised to find out how many breast cancer cell lines are out there that have ERB2 gene amplification. You're probably, if you follow this gene in the literature, you're probably used to looking at data for SKBR3 cells. That's a classic line that everybody works with when they're interested in ERB2. There's a couple of others that you might have seen in the literature, GIMT1, but maybe you're not as familiar with all of these other lines that have ERB2 amplifications. It's interesting that one of the things I discovered in working on this is that this line, AU565, is actually derived from the same patient from which SKBR3 came from. So you'll notice that their genomic features are quite similar. Okay, so now we've got the cell lines rank ordered by amplification and overexpression. Let's now look at what happens with regard to the essentiality scores. Well, the first thing you see, and this is quite a surprise, and we'll talk more about this later, but HCC 1954 just jumps out at you right away as an outlier. High level amplification, high level overexpression, not a hit in the screen. Okay, that's a surprise. Are we going to see that very often? I don't know. Well, let's look. 1569, also not a hit in the screen. CR 7530, yes, a hit in the screen. A hit in the screen, yes, no. These are close, right? This is close to that -0.5. SKBR3, a hit in the screen. And so now here, unlike the estrogen receptor, where there was this very tight correlation between the genomic feature of overexpression and essentiality, that association here is a lot more loose. And we're going to have a whole separate article and blog post with data on that. But this shows you right away that ERBB2 and ESR1 behave differently in this context. So some of these cell lines, ERBB2 is in fact quite essential in association with amplification, but not all of them. If we now rank order by RNAi score, we can bring up a couple cell lines that have amplification and essentiality, but these gene looks kind of essential, even though the gene's not amplified here, but it's slightly overexpressed. And of course, here it's amplified and overexpressed. So again, the picture with regard to ERBB2 is not nearly as clear cut. And so if you're interested in finding and working with cell lines that have an ERBB2 amplification and overexpression, and the gene is highly essential, this is basically where you'd start. You would start by this list and you would identify those cell lines for which the essentiality goes along with the genomic feature. And then if you're interested in the discordance, you would know which cell lines to pick. And again, GIMT1 is sort of a classic here. And because we know from the literature that GIMT1 is actually a breast cancer cell line with an ERBB2 gene amplification, but are resistant to ERBB2 targeted drugs. And knowing that, you're not too surprised to find that ERBB2 is not a hit in the screen in this cell line. That is, ERBB2 is not essential, even though it's amplified and overexpressed. So it's not essential. You might not be surprised to find out that it's drug resistant, and it is. And so here's where you can do a little bit more complex analysis of essentiality data in association with the genomic features of genes you're interested in. Let's now do, just very quickly, another gene that I've been interested in for a long time, the epidermal growth factor receptor. So we all know that EGFR tends to be overexpressed in basal or triple negative breast cancers and underexpressed in ER-positive breast cancer. So we can type in EGFR, and if you just look what comes up by the RNA, the genome scale essentiality screen scores, you see the gene isn't essential in the gene isn't essential in any cell line except for some 149. Now that's really interesting because we found it to be essential in our screen, and the Han lab found it to be essential in their screen, but no other breast cancer cell line is EGFR found to be essential, even though if you rank order by log fold change, there's a bunch of cell lines that overexpress it. And of course, MDA468 is a famous one because here the gene's amplified, overexpressed, and didn't do anything in either the CRISPR or the RNAi screen. BT20 is another famous line with amplification, overexpression, not a hit in the screen, and so forth. So again, this is very interesting and shouldn't be too surprising because even though we know that a lot of breast cancer cells overexpress EGFR at the message and protein level, we know that because of this, it was considered a gene target or a drug target for triple negative breast cancer. And we know that the drugs that target EGFR failed in clinical trials. I would argue that this is predictive of that, or at least consistent with that, because if the gene isn't essential, regardless of its genomic alteration, it's unlikely to be a good drug target. So ESR1 is a terrific drug target. ERBB2 is a pretty good drug target. EGFR, not a drug target at all. And that all goes along with what has been found clinically. So speaking of that, let's look at another very interesting breast cancer oncogene, the PI3 kinase, PIK3CA. This gene codes for the alpha isoform of class 1 phosphatidylinositol 3 kinases. Very specific. This is the only gene in the PI3 kinase family for which we find point mutations in breast cancer and other cancers, suggesting that it is a key signaling hub in breast cancer patients and cell lines. And I picked this one because I want to show you that not only does our database and tool identify amplifications and overexpressions, but also point mutations. And so if there is a point mutation in a gene that you're interested in that you type in here, and if that point mutation is represented at almost any level in the cosmic database, that is the catalog of somatic mutations in cancer database, it will be listed here. If there's a mutation that's not recurrent and has only been seen 1 or 2 times in cosmic, it will not appear here. So we didn't want to sort of overwhelm people with mutations that may have very little biological significance. So we only show the mutations that are common. This H1047R mutation is the most common PI3 kinase mutation in breast cancer. It's found many times in cosmic, and there's a range, some more or less. There's some different mutations. And again, you can rank order by finding the breast cancer cell lines with and without mutations. Now you have the opportunity to look at the association between this genomic feature, point mutation, and gene essentiality. Now you see something that looks a little bit more like the estrogen receptor. In breast cancer cell lines where there is a point mutation, particularly the common ones, both the RNAi data and the CRISPR data show good essentiality associated with these point mutations. It's not perfect, but it's strong. And again, we'll have a separate blog analyzing these data coming up another time. So again, if we rank order now by the CRISPR score, you'll see that cell lines that are most reliant on this particular gene, it's highly enriched for cells with the PI3 kinase mutations. And as again, I'll show you later, but I'll just give you a little hint. There are cell lines here like SKBR3 that do not have a point mutation in PIK3CA. This is an ERB2 amplified cell line. And in fact, the gene is quite essential. And just to give you a little hint of what's coming, this is actually quite common. There are many breast cancer cell lines with ERB2 amplifications and wild type PIK3CA for which PIK3CA is essential. And when you take them together, the ERB2 amplifieds or the PIK3CA mutants, those make up essentially all of the cells for which the PIK3CA gene is essential. So this is the kind of thing you can sort of mine out of these data. You can identify these surprising associations. I certainly didn't know this. I didn't know that ERB2 amplified cell lines, in the context of wild type PI3 kinase, that PIK3CA gene would be essential. And whereas in other cell lines that have neither ERB2 or PIK3CA mutations, this gene is not essential. And so again, it shows the power of the analysis that one can do. So again, you can identify genes that have point mutations, and you can see how common they are, and you can see how they're associated with essentiality. And as you would expect, genes with point mutations that are oncogenes activated by point mutations do not have changes in expression at the message level, nor do you find them with gene amplification. Okay, so again, there's an example of a gene that you can identify with point mutations. And I just want to, just to give you a sense of this, any gene that the gene name for, you can just type it in here. So, if you're interested in Wnt5A, you can type in Wnt5A. And you can pull up the data and you can rank order by relative expression. And you can find cells that highly overexpress Wnt5A at the top of the list, and cell lines that have no expression of this gene at all. The same is true for PTEN. This is really an interesting one, right? Because PTEN is sometimes mutant, as shown here, and sometimes those mutations are associated with a complete lack of expression, but sometimes the gene is actually disrupted in a way where it's wild type, but there's no message at all. And so there's essentially no expression of the gene at the message level. So you can identify all of these right away by looking at mutations versus expression and so forth. Now, in the case of tumor suppressor genes like PTEN and TP53, the Achilles, the screen data really don't mean anything because we can't really identify tumor suppressor genes by knocking them out in this sort of straightforward version of the screen. So here you're really mostly interested in mutations and relative expression. But again, any gene, and I have to say, I use this tool a lot when I'm listening to a seminar or reading a paper, and I'm listening to someone talk about this gene that they're now interested in, and they tell me that this gene is highly essential and it's overexpressed and blah blah blah. And so I just get out my phone and basically type in the gene name, and I pull up the data, and I look at the data in the database while I'm listening to the talk. And sometimes what I find is consistent with what the speaker is saying, and sometimes not so much. So it's a fun way to use this tool, actually, both when reading papers and listening to talks. Okay, so I just want to close by going now to the data mining tools for the sum lines. And again, in the last video, I talked about these different sets of tools, and go to the sum line gene query. And I'm just going to do this to show you, to highlight one key difference in the way these tools work here, because as I mentioned in the previous video, the method that we use to do the screen in my lab, even though the overall method is the same as what the Han lab use, our methods for quantifying the data are different. And so you don't find a series score here. What you'll find for our screen is quant log and rank score. So just to give you an example, I'm just going to now go ahead and do EGFR again here. And one of the things that's interesting is, several of our cell lines actually overexpress EGFR. And now here's sum 149 again. And so here, the Han lab data and our data are concordant in that, again, high level overexpression of EGFR. And this gene was a hit in our screen. And so when you're looking at the screen data for our genes, pay attention to the quant log rank, because basically we, again, rank ordered the genes. The lower the number, that is, the higher the rank in the screen, the more essential the gene was. And we typically identified around 1,000 screen hits per cell line. And again, if you look at that PowerPoint video or that the PowerPoint presentation I alluded to earlier, you can see exactly how we quantified this. But the bottom line here is, in our screen, we basically take the top 5% of genes that have hit scores that are sort of outside the normal be expected based on a normal distribution. That typically results in about 1,000 genes. And so if you just now go by quant log rank, you can see that, like for MCF10A, which we've known forever, EGFR is really important. It technically wasn't a hit in the screen because we only identified in this screen 753 hits based on that top 5%. But a rank of 753 is pretty good, and really is suggestive that this gene is a hit in MCF10A as we'd expect. Sum 102, again, as we published, the cell line overexpresses EGFR. 1079, it's pretty darn close to that cutoff. In this case, we had more genes that were hits in the screen, so at sum 185. At rank 1250, it was considered a hit because that was in the top 5%. So what I suggest in looking at the query data for the sum lines is you very much pay attention to the quant log rank, and you can make your own decisions about, when you start getting down here to 3,000, 4,000, 8,000, I mean, these are pretty dead, pretty negative, and it's not surprising that sum 44, which doesn't express the EGFR, is at the bottom of the list, 10,000. Sum 190, which doesn't express EGFR, is down on the list, 12,900, and so forth. Okay, so that's just one difference. The other difference here is that in the gene query and the sum lines, we also show the available drugs for some of these genes that are hits in the screen. And so, for example, there's another gene that we've worked on called BCL2L1 that we're interested in, which has a series of drugs that target it. And you can, again, find the cell lines for which this gene was a hit in the screen. Okay, so that's basically a Cook's tour of the gene query tool. Again, this is a really fun tool to help you gather a lot of new information about breast cancer cell lines you work with, and it helps you when you're learning about new genes and you want to understand the relevance of new genes, or even if you're interested in a new gene and you want to know what cell line to work with. I see this often where people become interested in a gene and they just use the cell lines they have in their lab. And they really don't have any idea how to find the cell line for which that gene might be highly important or not important at all. And so this tool can allow you to actually pick, a series of cell lines where the gene is expressed at different levels, have different levels of essentiality, certain mutations are not. And that way you can pick model cell lines for specific genes that are best suited for the questions you want to ask. So play with this tool. It's really fun. If you have any questions further about it, I'll be happy to do one of these Zoom meetings with you one-on-one and talk more about it. But like I said, this is a tool that I use all the time, and I really have a lot of fun with it. Okay, so thanks for tuning in. The next video that I'm going to do is going to be about the KEGG pathway engine, and that really is a blast to use. And I use it commonly, and I've learned a lot. So stay tuned and watch for the next video on the use of the KEGG pathway engine. Thanks, and bye for now.